When most people think about hospitals, they think of them as places where the sick go for treatment and healing. However, hospitals are also the most likely place that a patient or visitor could contract a microbial pathogen. Hospital-acquired infections are a serious threat to patient outcomes and pose a significant public health concern as we move into what many have dubbed the post-antibiotic era. Hospital design, in tandem with consideration for the natural evolution of microbial communities, could help us build better hospitals and reduce infection rates. There has not been enough research or systematic analysis of microbial communities and hospitals, but a group of researchers at the University of Chicago is working to change that with the Hospital Microbiome Project. To better understand the fundamental concepts behind the project, let's take a step back into the past for a quick lesson on the history of sterilization in medicine.
In ancient times, infections were considered the work of magical or demonic forces. However, the Egyptians began using medical practices in 3000 BC. Along with these practices came the first acts of sterilization using tar and pitch. Crude methods of sterilization remained for centuries, and most doctors dismissed the need for a clean medical environment. This changed with the introduction of the modern era of medicine. Ignaz Semmelweis proposed in 1847 the need for hand washing of doctors. However, his theory was widely dismissed by his peers until Louis Pasteur published his germ theory. Joseph Lister, the father of antiseptic surgery, furthered the cause of sterilization by dipping surgical rags in carbolic acid and using it to sterilize his instruments and hands. New methods quickly emerged, and in 1881, boiling became a leading method of purification. Gustav Neuber created the first antiseptic operating room, thus ending the traditional large operating theaters. Dr. William Stewart Halsted made gloves a commonplace in the operating room. Sterilization units were developed to purify surgical tools. These units have used steam, irradiation, ethylene oxide, and eventually advanced plasma technology in the 1990s and until now. Ethylene oxide is now the most universally used chemical, which sterilizes syringes, instruments, masks, catheters, along with a multitude of other hospital equipment.
All of these breakthroughs in sterilization techniques drastically reduced the infection and mortality rates in hospitals. People were no longer dying as frequently from simple procedures or surgeries. But we took our war against microbes a step too far, and the microbes have begun fighting back, with the power of natural selection and evolution on their side.
The overuse of antibacterial cleaning products, including disinfectants in the home and in hospitals, may be producing strains of antibiotic-resistant bacteria. When a population of bacteria is attacked by an antibacterial chemical, a smaller population armed with special defense mechanisms can develop. These populations of bacteria can survive and reproduce as their weaker ancestors decease. Antibacterial chemicals select for bacteria that can continue to live on in their presence. As bacteria begin to develop a tolerance for these compounds, they are also developing a tolerance for certain antibiotics. Resistant strains of bacteria can lead to increased infection risk in the community, hospitals, and other healthcare settings.
So we now know that our overzealous efforts to eliminate germs from our lives may be doing more harm than good. Understanding microbial ecosystems and how we as humans impact them is a core component of the Hospital Microbiome Project. But this concept started small, with the Human Microbiome Project.
A microbiome is defined as the microorganisms in a particular environment. These environments can include nearly every aspect that maintains life, such as plant life, the human body, or an environment. A recent study in the United States was funded by the National Institute of Health, known as the Human Microbiome Project. Conducted over 4 years, results of the project were published in 2012. The purpose of this study was to compare and identify the various microorganisms in the body of both healthy and diseased individuals. Researchers used whole genome sequencing to gain a comparative perspective on the different individuals' species. 5 body types were isolated: oral, skin, female reproductive organ, and nasal and lung. Furthermore, the project looked into the bacterial 16S rRNA sequences by amplifying the polymerase chain reaction from the subjects. By studying the certain types of bacteria, the metabolic genomic approaches could be used to identify potential viral diseases.
The Hospital Genome Project was conducted at the University of Chicago Medical Center. Researchers sampled 2 patient rooms, 2 nursing stations, the water supply, and air filters over the course of a year. The patients were swabbed on the hand and in the nose. Room surface samples included floors, bed rails, and glove boxes. Nursing stations were sampled weekly on surfaces such as the countertops, computer mouses, phone handles, and chairs. Once the samples were collected, they were processed in a variety of ways in order to identify and understand the microbes that grow in hospital environments. First, the 16S, 18S, and ITS genes were amplified using PCR. CHIME was used to phylogenetically classify microbial species and determine alpha and beta diversity in the samples. Source Tracker allowed researchers to identify what portion of microbes originate from each location. Finally, location similarity analysis was used to identify patterns of microbial growth, which aids in the prediction of which organisms will grow in a specified area.
The end goal of this massive collection and processing of data is to track the changes in development of the hospital microbiome. Lead project researcher Dr. Jack Gilbert hopes to be able to answer questions about where infection-causing pathogens are originating from, how they travel within hospitals, and how humans are actively shaping the microbial character of the hospital itself. A critical observation that other microbiome researchers have made is that there is a stark contrast between the microbial community inside of a hospital and the communities just outside its walls. Inside an air-conditioned hospital room, a disproportionate number of typically rare pathogenic species can be found. This means that patients are effectively steeping in their own deadly bacteria and viruses. Just outside the hospital walls, the air is filled with a diverse array of harmless microbes from the plants and soil. The hyper-sterilized environment of the hospital cuts down on the microbial diversity, eliminating harmless bacteria that would normally impede the rapid growth of pathogenic species. In short, we may be creating more dangerous microbial ecosystems for some of our most vulnerable populations. Addressing this dysbiosis in hospital microbiomes is a goal for the future. There is still much research to be done before we will be able to use building design to craft more balanced microbial ecosystems. But this idea is already gaining speed in the architecture and design community. By restoring microbial diversity in our buildings, maybe we can cut down on the deadly infections that continue to haunt doctors and patients alike. Operating rooms and intensive care units are a few of the areas in hospitals that will always need to be sterile, and there are other valid concerns about the safety of manipulating hospital bacterial communities. Nevertheless, Jack Gilbert and the rest of the hospital microbiome team are optimistic about the future of our relationship with microbes, a future in which we work with the microbes around us and within us, instead of just against them.